Hello everybody, welcome back to Dr. Sam's Anatomy classes. Myself is Dr. Azmi Musin and I'll be covering up this topic, Jacobson's organ, that's also called vomeronasal organ. This is somewhat related to the olfactory system, and if you remember in the olfactory system, when I was teaching you about the olfactory pathway, there I mentioned that this sense of olfaction, the sense of smell, is more developed in the lower animals, in the terrestrial animals like reptiles and other quadrupeds, mammals also. Now similarly, this Jacobson's organ is a receptor site at the vomeronasal organ, and this is carrying the sensation of smell, the specific smell from the pheromones. But the pathway is not that of the olfactory nerve. It is a secondary pathway, and that pathway is called accessory olfactory pathway. It uses the pathway differently from the olfactory pathway. So that's why it's called accessory olfactory pathway. An important thing is that if you're not aware of it, there is a cranial nerve which is said to be zero cranial nerve. The zero cranial nerve is this nerve which carries this sense of specific smell from the Jacobson's organ, and this is actually taking origin even more rostral to the first cranial nerve, and that's why that nerve is called zero cranial nerve. And that relays its sensations, the sensory neurons relate sensations there in the accessory olfactory bulb. Further, the details about its pathway I'll be teaching you in this lecture. And remember, one more important, interesting fact about this pathway of accessory olfactory pathway is, remember olfactory pathway, while I was teaching you, I told you that that was the only sensation, the sensation of smell, which bypasses the thalamus to reach the cerebral cortex. The rest, all of these sensations may be general sensations or maybe special sensations. They all reach the cerebral cortex. Why the thalamus or the similar nuclei adjacent to thalamus, like, retrolentiform nucleus, sublentiform nucleus, they were being used by the optic and auditory pathway also. But olfactory pathway was directly reaching to the cerebral cortex, bypassing the thalamus. Now coming to this zero cranial nerve, which is carrying sensations from the Jacobson's organ, this even bypasses the thalamus and the cerebral cortex both. This is an important point. Okay, so the sensations from this organ are being carried by zero cranial nerve, and that sensations reaches directly to the limbic system, and from there to the hypothalamus. So it bypasses both the thalamus and the cerebral cortex. So these were some important points about this pathway. Now, this Jacobson's organ, the purpose mainly is for getting attracted to the opposite partner for sexual gratification, for sexual intercourse. For mating partners, this is an organ which, liberates these pheromones, and the other partner after smelling gets, aroused for the sexual activity. While in lower animals, there are other, purposes is like for craving for food, like, targeting the prey with this smell, and for saving for life also. If the smell, is from the predators, the lower animals smell it and they escape for their life. So there are three important functions about this Jacobson's organ. Okay. So let's begin about this organ. Let me tell you, this Jacobson's organ is named after the great scientist who first explored about this organ and mentioned about it in its book. And he was a Danish anatomist in 1811. The scientist Ludwig Levin Jacobson. He was the scientist who discovered this, and that's why it's after his name. We know this vomeronasal organ as Jacobson's organ. It's an organ of chemoreception because it perceives the moisture air particles or even directly touching to the wet particles. So it's an organ of chemoreception that is a part of the olfactory system of amphibians mainly. Remember, it's part of amphibians, reptiles, mammals. Although it does not occur in all tetrapods. So this should also be known that it's not a general feature in all mammals, rather it's only peculiar to few of the tetrapods. Olfactory epithelium, if you remember, that was forming the roof of the nasal cavity around 2 to 2.5 centimeters square in the one side on the nasal chamber. But remember, this Jacobson's organ is not a part of olfactory epithelium. because olfactory epithelium lies in the upper one-third of the nasal chamber, and this thing is not present there, rather it is present on the nasal septum in the anterior, lower one-third of the nasal septum. If you remember the nasal septum, where this anterior margin of the vomer attaches to the septal cartilage, there you find this organ in humans, right? But in lower animals, in reptiles and in some mammals also, it is even present in the hard palate. So they even smell with their mouths open in such cases. Okay, so it's a patch of sensory cells within the main nasal chamber that detects heavy moisture-borne odor particles. Now you see here, that's how this organ detects this pheromones or the other chemicals, the smell from prey or from the predators. So the rats also have this in the antero-inferiorly in the nasal chamber. Even the elephants, this is the proboscis of the trunk, and if you've seen the ends of the nostril of the trunk of elephants, they have a pointed projection at one end, and that's called the finger of the trunk of the elephant, and it's through this, it licks the liquids from the opposite sex, and then it brings its tip of the nose towards its oral cavity, and where in the hard palate or in the anterior nasal chamber you have this organ, so it smells with the help of its proboscis or the trunk. You can see here there are other mammals which have the vomeronasal organ at the tip of the hard palate anteriorly just below the nose, and there are canine animals like even the cats, they have their vomeronasal organ just above on the floor of the nasal cavity, that's hard palate. And you must have seen one thing is panting. Panting is like, when animals bring out the tongue, that is, for hyperventilation sort of thing. But this feature, this gesture you're seeing in the horses or the donkeys and all, where you find them, they open their mouth and they extend their neck, and then they open their mouth, they lift their upper lip or they uproll the upper lip, and they expose their teeth, and in doing so they are actually withholding the breath, and at the same time they are squeezing this vomeronasal organ present in the anterior nasal chamber, where they are actually squeezing. Actually it's a fluid filled, it's a semi-coiled crescentic type of tubular gland, which there is already fluid filled there, so they squeeze that gland, and when after that then they inhale the smell. They try to bring the vomeronasal organ more closer to the air in front, so they expose the vomeronasal organ in doing this gesture, so that they can inhale the smell which is actually interesting them. And this maneuver which they're doing is called Flehmen response. Similarly is here, you can see this called Flehmen response. When they lift their upper lips, they uproll their upper lips and they bring out the tongue and they open the teeth. Some animals, they have their organ within the hard palate. So that way, they are actually either they are attracting, they're getting attracted or getting prepared for the sexual activity, or they are becoming aggressive to chase their prey. Right, look here, this is the snake. Can you see here, this is the forebrain, and from the forebrain this is the nerve which is connected to this Jacobson's organ. So have you seen the snake or even the lizards, they bring out their tongue, right? They have the bifided tongue and they rub their tongue against the wet substance, and after getting their tongue wet or moistened with the chemicals of the surrounding, they withdraw their tongue partially, so that the moistened tongue with the chemicals which have been absorbed on their tongue, they can bring it closer to this Jacobson's organ, through which they can smell the surrounding liquids or the prey to which they are targeting to. So that was about different animals, how this is present there. Look here now, in reptiles or in lizards or salamanders, I was telling, the process of chemoreception using the Jacobson's or vomeronasal organ, they have explained it here. You've seen it, like this is the olfactory bulb, right, from the forebrain. This is the nerve which is attached to this Jacobson's organ in the anterior nasal cavity, or you can say at the roof of the hard palate, which has this opening towards the base of the floor of the nasal chamber. So at the same time they protrude out their tongue. When they protrude out the tongue, that is an extended tongue, the odor particles are captured from the air by the tongue. So the tongue, when gets moistened with the chemicals inborn in the air surrounding, then they pull up, they retract the tongue. When they retract this tongue, the odor particles mixed with the fluids in the mouth are delivered to the ducts leading to the vomeronasal organ. As you can see here that these vomeronasal canals, they're opening in the roof of the buccal cavity. So that's how the snakes get to know about the food they want to eat, or they can chase their sex or the mating partners. Okay. Now vomeronasal organ in human beings. That's what I was telling. It's this olfactory epithelium. I've already told you this is occupying the upper 1/3 of the nasal chamber or the roof of the nasal cavity or the undersurface of the cribriform palate. But the vomeronasal organ, it is present anteroinferiorly on the two sides of the nasal septum. You can see it's this vomer and this septal cartilage. That's this point where you find is the vomeronasal organ. It's very small and it's like a patch of specialized epithelial cells at this place, which is a tubular type of concentric tubular type of gland. And this is how, the histological features you can see here in the Jacobson's organs, very much similar, like, the olfactory epithelium. There were three types of cells. Even here, there are three types of cells. One is the, receptor cells. Receptor cells are actually, again, just like, remember the first cranial nerve. I said that the first order sensory neurons in the first cranial nerve, they are actually behaving as the receptor as well, because their peripheral receptors are like the dendrites embedded in the nasal mucosa, while the cytons lay outside the, in the nasal mucosa, while the central processes pierce the cribriform palate. Remember what I'm telling is about the olfactory epithelium. It's a revision. So there are like, 16 to 20 million neurons in the nasal mucosa, all of them in thousands of groups. They bundle up with their central processes passing to the cribriform palate, and then they form around 16 to 20 phyla olfactoria. That's also called the olfactory nerve. So 20 nerves on each side, separately on the two sides you have 20 nerves which reach to the olfactory bulb, where they relay with the mitral or the ducted cells. But there were also inhibitory neurons. Remember, in the olfactory bulb, they were like, granule cells and paraglomerular cells, periglomerulocytes. So they were also inhibitory neurons. Now here, this, tubular duct-shaped organ, the Jacobson's organ, where you find their cells, basically this organ has been, not very much studied in humans. So they mention it as dark cells and light cells, dark cells and light cells, and on the base, you finding are the basal cells. Now basal cells are of course the progenitor cells for the replacement of those cells being shed off. Now the dark cells, which are actually the receptor cells, these receptor cells on the receptor side, they have microvilli, and onto the microvilli, they have a G protein coupled receptors. G protein coupled receptors, and they identify the pheromones, the chemicals which they get attached to it, and then their processes. That chemical thing is being processed into the, impulses in the or action potential in the neurons, and is being carried by the zero cranial nerve to the accessory olfactory bulb. Okay, and while these supporting cells, the light cells are the supporting cells, and the basal cells are for the replacement of the fallen or the dying out cells. That is about the Jacobson's organ. Now accessory olfactory pathway or the zero cranial nerve. So this I'll try to draw it for you so that it becomes easy. Look here, actually it's a concentric type of organ anteroinferiorly on the either side of the nasal septum at the junction of vomer and the septal cartilage. And this has a notch, this opening which is directed downwards. This opening is downwards. And as I have already told you that there will be cells on the inner surface, these are the cells. There are light cells, dark cells, receptor cells, then there will be the basal cells, right? So it's like, it remains stuffed with filled with the fluids already secreted, and because of a lot of mucosal glands surround this gland, and of course the blood vessels also surround it. So there are actually two pair of these glands on the two sides. They are separated, the two pairs, they are separated by the nasal septal cartilage. So they are two pairs suspended on the two sides. And this is vomeronasal organ. Now these cells, they have the cell bodies within this, right? And the peripheral receptors are here, peripheral dendrites. The central processes ascend up and they reach into the accessory olfactory bulb. Now this is accessory olfactory bulb. And important thing is that this nerve is zero cranial nerve. The pattern will be the same like, first cranial nerve, because these are first order sensory neurons. These are bipolar cells, as I already mentioned, that they have the cytons lying here. So these are bipolar cells with their peripheral dendrites acting as the receptor side, which have G coupled protein receptors. The central processes, they form the zero cranial nerve, which relay in the accessory olfactory bulb. Well, this nerve, zero cranial nerve, have also been mentioned by some authors as the terminal nerve. It is also mentioned as terminal cranial nerve. Then from this accessory olfactory bulb, if you remember the first cranial nerve, this was continuing as olfactory tract and was having lateral and medial olfactory stria. They're all being separate from the olfactory trigone. But here, these efferents from this olfactory bulb, let me draw it here. This is like amygdala. This is the bed nucleus of the stria terminalis. So this is bed nucleus of stria terminalis, and this one is amygdala. Okay, this is amygdala. This was the first order neuron. So the second order neurons from here will relay in the amygdala, or they might reach into the basal nucleus of stria terminalis. There's some neurons which might reach from amygdala to the basal nucleus of stria terminalis. And destination, the last where it will reach, is called the hypothalamus. So the third order neurons from this basal nucleus stria terminalis as well as from amygdala, they will relay here in the hypothalamus. So you've seen that this is the accessory olfactory pathway, where the pathway has bypassed both the thalamus and the cerebral cortex. Okay, now because hypothalamus is a major neuroendocrine center, so in response to this stimulus, it starts secreting gonadotrophins. And it's this response through the gonadotrophins, neuroendocrine system, that the animal or the person starts behaving according to the hormones being liberated. So that is about the accessory olfactory pathway. Now basically, why this zero cranial nerve? Basically, let me tell you, like during the embryological development, as ontogeny repeats phylogeny, even if adult human beings may not be having it present, this vomeronasal organ, but during the course of development, each human embryo behaves or develops through all the evolutionary stages. So during the development, and the infant or the newborns and infants, they do have this completely developed vomeronasal organ or the Jacobson's organ present in them. And even in young adults, this power of being attracted to the pheromones, or liking the smell, being attracted to the pheromones of the body odors, is also seen in early in the adult or the childhood age group than in young adults. And as with age, this power also keeps on decreasing. So if you remember, because the nose that develops from the nasal placodes, you must have been taught about the development of face, right? So initially there were nasal placodes being developed. At that point where the nasal placodes were being developed, there is these cranial nerves that are formed from the anterior most edge of the neural plate. So it is even more anteriorly in development to the first cranial nerve. Because lately the nerve being studied by the modern scientists, they've classified into 12 nerves. But since 1811, the scientist Jacobson, who discovered this, like he didn't went into the details about this pathway, but lately after this discovery of the 12 nerves, scientists probed upon this and then they found out this nerve. So that's why this nerve has been named as zero cranial nerve. Right now, about this substance, it's about the liquids. Some animals, they dip their tongue, as I told you, in case of snakes or reptiles, then they bring their tongue close to this organ. Some animals like elephants, they dip their proboscis to the tip of the trunk and then they bring the trunk close to the organ. And this, let me tell you, is mainly the urine sample from where they detect these chemicals which they smell, even in reptiles, and from where they can chase the animal to targeting them as their prey or their sex partner. Now about the response, the behavior from here. The behavior here will be an appropriate behavior according to the type of smell that this Jacobson's organ or VNO perceives. If after smelling they perceive that this smell is from something tasty to eat, right? So that animal's thing will be taken as a prey. So that animal will chase that animal which is targeting for its desire to fulfill the hunger. And if this smell is of some bigger animal from the predator animal, so this animal which is smelling it, he might find a way to escape to save its life. And the third response is like if the smell or the pheromones which it inhales, if that excites or arouses him for sexual arousal, then the animal will behave accordingly for sexual response. But the animal targeted should be a potential sexual partner. Basically, finding a sexual partner with the help of smell is actually the mate partner should be of the same species, right? Actually, it makes an option, or the animal selects the animal, the sex partner according to the smell. If it's interesting, if it's arousing, then only he chases other animal for this sexual intercourse. Okay. Flemen's response. You can see and observe what's happening here. Animals usually do it in response to the olfactory stimulus, although it has been seen that the quadrupeds, mainly the ungulates, that's like horses, donkeys, deer, zebras, giraffe, all these animals, they enjoy doing this Flehmen response. Might also be due to some visual response sometimes, but mainly it is because of the olfactory response that they perceive. Look here now, that gratifies is actually the smell of urine of the opposite sex partner. So when they inhale it, you can see the response that you find is that they elevate the neck. What actually Flehmen means, like exposing the upper teeth. So Flehmen means exposing the upper teeth, like this way. So animal actually extends its neck and then it uprolls its upper lip, exposing the teeth, and of course exposing the oral aperture. While at this moment it actually is closing its nose, its nostrils, and is trying to inhale through its oral opening, right? So this response is actually so as to expose the Jacobson's organ. Jacobson's organ normally, as I told you, in the ducts of this gland opens into the hard palate and just behind the upper incisors. So when it does like this, is actually bringing the Jacobson's organ more close to the air surrounding, and in doing so it inhales at the moment. Actually it holds the breathing, it withholds the breathing for a few seconds, and then he inhales or engulfs a big amount of that air which is arousing. That is actually the Flemen's response. However, in some quadrupeds you might find it in a different way. They are actually extending the neck and the head and lifting it high up in the air and doing the same maneuver. So that is in response to their sexual gratification, and that is called Flemen's response. Now see what humans have done. This is what I was describing as Flemen's response. Now they have prevented the animals from doing this thing. They have made these green guards. The green guards, then when they wear it, actually it's a sieve like thing, so it provides easy breathing, but it actually limits the eating of the grass, and that is called nibbling, just like superficially biting the grass. So actually this is called the muzzle area, or nasal and oral opening together is called the muzzle area. So they wear this mask onto this muzzle area, and actually it's not limiting the breathing, but it's limiting the grass intake, and of course it is limiting the Flemen's manure, the Flemen's response. Okay, so it's done. Thank you.